Today we'll be talking about the lac operon negative regulation. These are going to be our learning outcomes.
Before we discuss the negative regulation of a lac operon, it's important to know what an operon actually is. An operon is just a group of genes with a single promoter and is transcribed as a single messenger RNA. We're focusing specifically on lac operons, which contain genes that encode proteins that allow bacteria to use lactose as an energy source. Even though bacteria can break down lactose with a lac operon, it's not a preferred source of energy. Glucose is a better source of energy because it takes fewer steps and therefore less energy for bacteria to break it down. So if glucose is present, bacteria is going to use it. If lactose is the only sugar present, however, bacteria will break it down, which is why the lac operon is so useful. In order to break it down, the lac operon genes must be expressed, which encode critical enzymes for lactose uptake and metabolism. To be as efficient as possible, E. coli should only express the lac operon only when lactose is available and glucose is not available. Glucose and lactose levels are detected by two regulatory proteins. The lac repressor, which will be discussed later in the video, acts as a lactose sensor, and the catabolite activator protein acts as a glucose sensor. These proteins bind to the DNA of the lac operon and regulate its transcription based on lactose and glucose levels.
Let's take a look at the structure of the lac operon. The lac operon contains 3 genes and several regulatory sequences. These work together to express the genes when glucose is not present, but lactose is. LAC-Z codes for an enzyme that breaks the glycoside bond, which breaks down lactose into monosaccharides so that it can be used throughout the cell. LAC-Y codes for a transporter protein that will bring the lactose into the cell, and LAC-A codes for a protein that will essentially take care of the beta-galactosides that can't be broken down, which eliminates toxic compounds from building up in the cell. These genes are controlled by regulatory DNA. Now, regulatory DNA are simply regions of DNA to which proteins can bind, controlling the transcription of the operon. In this case, they interfere with the binding site for RNA polymerase. The operator is the place where the repressive proteins will bind. Essentially, it overlaps with the promoter so that when the lac repressor protein binds to the operator, it blocks RNA polymerase from binding to the promoter and transcribing the operon. When lactose isn't present, the repressor binds to the site, preventing translation. The other piece of regulatory DNA is the cap binding site. Now, this is important because when it is bound to the DNA, it helps the RNA polymerase bind as well, which in turn increases transcription. So when we look at the picture on this slide, whenever lactose is unavailable, we have a repressor protein prohibiting transcription. However, when there's low glucose, we see that the cap protein is then present. This allows for the cell to only produce these genes when glucose isn't available, but lactose is. Otherwise, the genes either aren't expressed or are expressed at a very low amount.
So before moving on, a negative regulation is the binding of a specific protein that inhibits transcription from occurring. In our case, we're going to use the lac repressor protein. So the lac repressor, as mentioned earlier, is a DNA binding protein. Its function is that it inhibits the expression of genes coding for proteins involved in the metabolism of lactose in bacteria. To answer the question of where does the lac repressor come from, we gotta look in a more detailed way. There's a gene that encodes for the lac repressor protein, and this gene is called lac-L gene. This gene is under control of its own promoter. The gene is near the lac operon, but it's not actually part of the operon itself. It's also expressed separately. Lac-L gene is also continually transcribed, so its protein product, the lac repressor, is always present. So in order for us to know how does it work, we gotta make sure first that we know if there's a lactose or no lactose. Because when lactose is not available, the lac repressor binds tightly to the operator, preventing transcription by RNA polymerase. But when lactose is present, small allolactose molecules bind to the lac repressor and makes it change shape so it can no longer bind to the DNA. So this change in shape makes it floss off the operator, clearing the way for the RNA polymerase to transcribe the operon. Just a side note here, the lac operon is considered an inducible operon because it's usually turned off or repressed, but can be turned on in the presence of the inducer allolactose. So I'm just going to review the key points or the take-home conclusion real quick. So the first one is that the lac operon of E. coli contains genes involved in lactose metabolism. It's expressed only when lactose is present and glucose is absent. The second key point is that there is two regulators turn the operon on and off in response to lactose and glucose levels. The first one is the lac repressor, which is the negative regulation, and the second one is CAP, which is the positive regulation. The last point is that the lac repressor acts as a lactose sensor. It normally blocks transcription of the operon but stops acting as a repressor when lactose is present. The lac repressor senses lactose indirectly through its isomer allolactose.